A couple of days ago, this is what I saw under my microscope. Thousands of something. Usually, at a glance, I have some initial idea of what I'm looking at, and this time was no different. My knee-jerk reaction was that this was bacteria, lots of bacteria. When water samples start to break down and begin to get a little smelly, just like the sample that I was looking at, it's no surprise to find huge quantities of bacteria. Here are examples of bacteria I've filmed in the past. Look familiar? But this seemed different. Something was gnawing at me, a sense that something was wrong. It was the way they moved, their speed, the distance they were covering. While I couldn't put my finger on why, in almost an instant I decided this wasn't bacteria. But if I was right, what was it? And so began 2 days of detective work to try to figure out what I was looking at. Micro mysteries. I've been at this microscope hobby for almost 3 years now, and I spend quite a bit of time viewing and imaging the life I find. For the most part, if I can't identify an organism myself, I at least have a fundamental idea of where to look to figure it out. Sure, every once in a while I see something new, like this thing, and I have absolutely no idea what it is. But this time, I was stumped. It was time to put on my detective hat. Learning what this really was wouldn't be a groundbreaking discovery. I certainly wasn't seeing something that has never been seen before. But I have a fundamental need to know. It's why I love science hobbies in the first place. The only place to begin is to review what I know. First, these organisms are incredibly small. The scene you're looking at is 3.5 millimeters wide by 2 millimeters tall. The tiny white dots are a fraction of a millimeter, a very small fraction. I also know that they tend to aggregate in areas where there is plentiful oxygen. I found them around air bubbles, at the edge of the slide where water met air, even around this colonial algae that is producing oxygen through photosynthesis. And after a few minutes, I could see large groups moving across the slide, most likely looking for oxygenated water after depleting the spot where they had been. When I magnified them a bit, I could see that many of them really fly around. They don't just bounce in place. And when they move, they're fast. They seem to cross vast distances in a heartbeat. Watch this one as I advance just one frame of video at a time. It travels dozens of times the length of its own body in just 3 frames. That's 1/20th of a second. I could also see that they had volume. I couldn't identify a specific structure, but they were clearly 3-dimensional. And as they passed in and out of focus, I could see they had dark inclusions, little dots that might be organelles or something they ate. At even higher magnification, I got a better sense of their shape. In fact, as I zoomed way in, much further than my optics could resolve, it was like I was looking at dice with the spots displaying numbers. Now, I have a pretty decent microscope, and while it was built 40 years ago, it's been a workhorse for decades in a university lab. But even with this microscope, it would be very rare for me to see such shape and detail in bacteria. Most bacteria are usually just too small. But if it wasn't bacteria, what was it? There were really only 2 options. It was either a ciliate or a flagellate. You're probably familiar with ciliates, such as paramecium. Tiny hairs, or cilia, covering its body, move in unison to propel it through its environment. Ciliates can move incredibly fast and cover great distances, a function of their low Reynolds number and the physics of fluid dynamics. I did a deep dive explaining this, and I'll leave a link to that video in the description. Flagellates, on the other hand, have one or more rope-like flagella, which they use to both propel themselves and grab food. The tiny flagellates I see usually move slowly, smoothly, methodically. I'd never seen them move like this. But I couldn't see either cilia or flagella due to the resolution limitations of my scope. I even have access to a special lighting technique called phase contrast, which normally would show me these details. Here are a couple of examples. But in this case, the artifacts resulting from so many organisms moving so quickly in all dimensions just created a blurry mess, and I couldn't resolve a thing. Based purely on speed, I was thinking these had to be ciliates. And while I couldn't resolve much detail, I could measure their size. They were about 3 or 4 microns in length. That's incredibly small. An average human hair can be 15 or 20 times that size. And this is where I hit my next roadblock. I asked Claude AI what size the smallest ciliates were, and it said 10 microns. That's 2 or 3 times bigger than what I was looking at. As anyone should, I didn't take that answer as gospel and did some follow-up research. Sure enough, there just aren't ciliates this small. That left only flagellates, unless of course I was wrong the entire time and these were in fact bacteria. I was running out of ideas, so my next step was to ask for help. I posted some video clips in a microscopy Discord frequented by a very diverse group of individuals. An initial reply pointed out that bacteria are a huge group of organisms, and I shouldn't dismiss that as the answer. It was a fair point. But while I realize I have seen only a fraction of the life on this planet through my microscope, I still felt like that wasn't it. While waiting for a reply, I went back to Claude, this time investigating the flagellate angle. Claude replied that they could be nanoflagellates, the smallest of the small. At the very same time, someone else chimed in on Discord that the movement just didn't look to them like how bacteria move, and they too thought it could be nanoflagellates. I tried imaging more slides to see if I could see any hint of a flagellum. No success. I could see what looked like some of the organisms dividing, a reproductive process called binary fission, and it was clear there were inclusions inside the cells, those tiny black spots, but nothing more. And so I was resigned to leave it there. I had a hypothesis, these were tiny flagellates, not bacteria, but no way I could get evidence of this with my existing technology. And then, another member of the Discord server made the key suggestion. Add a substance to the slide to thicken the water and slow these organisms down. Why hadn't I thought of that? I have a bag of methylcellulose powder which, when mixed with water, creates a thickening agent that's safe for microscopic life. A 3% solution, added to a drop of water from the sample, might just slow these things down enough to see a flagellum, if it's there. And here's the result. I haven't slowed down the video, it's real time, but the organisms are moving in slow motion because the water is a lot thicker. And there they were, flagella rotating like tiny propellers. I could see them everywhere. I finally had the evidence I needed and could confidently identify these organisms as flagellates, not bacteria, not ciliates. The mysteries of the microcosmos are never-ending. Hobbyists like me will always want the answers. And sometimes we may even get them. If you enjoy exploring the mysteries of the microcosmos, I hope you'll consider subscribing to my channel. If there's an interesting experiment you'd like to see, let me know in the comments. Thanks for watching. I'll see you soon.